Happy Friday and happy May Day. Before we get started, think about what is an independent variable? What is a dependent variable? When we talk about living things, there's actually some debate about how many kingdoms, how many groups of living things they are. But for the purpose of today, we're going to go with these six. So we've got plants, animals, fungi, protists, eubacteria, and archaea. Um, on this diagram, it looks like it says archaeobacteria, but actually they're not that similar to bacteria, so scientists just call them archaea. Again, if you look this up, over the past 10, 20 years, scientists have debated, are there 5 kingdoms, are there 6 kingdoms? So there's not a whole lot of agreement, even still, but that's kind of why science is so cool, because scientists are still discovering things and debating things. So a quick review of how we know things are living. All living things grow and develop, use energy, make waste, reproduce. They're made of similar chemicals, respond to their surroundings, and they're made of cells. So today is all about number 6, made of cells. One way to tell the difference between the different kingdoms is, are they made of one cell or are they made of many cells? So protists, bacteria, archaea, they're all made of one cell. Fungi, plants, and animals are multicells. They're more than one cell. And actually, fungi can either be one cell, like a yeast, or it can be many cells, like a mushroom, like you see here. In order to take a look at some of these cells, I used a microscope that I borrowed from school and some slides that were already prepared for me. This first slide you'll see is actually bacteria. That's in pink. It's been dyed to see easier. It's actually part of a larger colony. These are examples of protists, really tiny, dyed different colors to tell the difference. Now what I'm doing is I'm actually preparing a yeast slide, which is a fungi, and this is what yeast looks like under the microscope. Here I took a plant cell, a plant to see the cells on the microscope, and here they are, super bright green. Another plant, but underground plant, an onion. Under the microscope, really cool, you can see the cells. This is even closer up, you can see the cells really well. Then to use a cheek cell, a human cell, I took some cheek cells from the inside of my mouth, put them on a slide, and then if you zoom in, you can just see the outline of the cheek cells. They're kind of lumpy potatoes with a circle in the middle, and then if you dye them, this is what they look like. For the last one, this is actually human blood cells. You can see the red blood cells, and that dark one is actually a white blood cell. So let's take a little bit of notes. So make sure you have your notes sheet in the same spot as your notes from last time. The difference here is going to be between single-celled and multi-celled organisms. Now we're going to take a look at the cells because that's one way you can tell. Is it a protist? Is it a plant cell? So single-celled organisms are living things that are made of only one cell, single cell. Makes sense, right? So examples are bacteria, protists, and some fungi. So yeast, like I said, is just one cell. Multi-celled organisms are living things that are made of more than one cell. So like plants and animals, and then some fungi like mushrooms are multi-celled. And just a quick reminder, if you haven't heard the word organism, it just means a living thing. So because we don't have microscopes on hand, or at least many or most of you don't have microscopes on hand, you're going to do a simulation of a microscope. So you go to Gizmos, log in, and mine is already saved, but you can look at the Google Doc for your password and your username, and click on the one that says cell types. You can go into my classes first and then do that way. Either way, we're trying to find the gizmo that says cell types. So go ahead and launch that gizmo. So there's a couple of things to notice about this. So if you hover over the different things on the gizmo, it'll light up. But it might be easier to just go ahead and click show all samples, and then it says. So the first sample is going to be E. coli, a bacteria in the stomach of a cow. So you just click on it. It has a little picture. You're going to click on microscope. And then you can see there's these different dials. So the coarse focus you want to do first. So that kind of brings in the bacteria sample. So once I get it, there we go. And now to fine tune, you can do some fine focus until it's more crisp and clear, just like you would on a microscope. You can also do stage left or stage right to move it around. So we're on 400 magnification. You can go to 1000, 250. You can also show labels and maybe fine focus a little more so you can see better. And then you can also click on show information. All right, so this is a little bit away. You don't really need this last tab, so then you can go back to landscape, pick another sample. So just follow the steps in the Google Doc. It'll show you exactly what to do.